Okay, well, welcome. I'm glad you made it through the snow. Hope you get home safely tonight. Hopefully this stuff goes away. I'm ready for spring. I've seen my snow for the year. I'm happy, satisfied. If we were in Alabama right now, they'd be running for the store, getting batteries and milk and bread and water and hunkering down, and school would be canceled, and businesses would close, and Walmart would be the only thing open in town, and just a different perspective, isn't it? Remember that we have three things in front of us. We've got the first thing, take-home exam, Saturday at midnight. We've got the exam next Thursday, cardiovascular, blood, heart, and vessels. And then the Tuesday before Thanksgiving, the short paper. So those are the next three things in front of you. Then when we come back from Thanksgiving, guys, as you know, there's only one more week of classes. The week after Thanksgiving will be your lab exam and your last two lecture exams that week, and then we come back next week and all you have are final exams. So it's going to come really, really fast, and hang in there. The vocabulary on this next exam will go through 92. Let me pick up with about 5 of those slides right now. Then we'll finish up with the last little bit over blood. I think I owe you the platelet section, and then we'll go through the heart today. And that will leave us just with blood vessels next week. Well, I'm thinking about announcements too. Don't forget that your mastering assignments are posted, and you certainly will by the end of today have the material you need to conquer the blood homework assignment as well as the heart homework assignment. And I would really suggest that you not wait until after the weekend to start looking at mastering assignments. Try to knock off the 2 that we've done, and then that would leave you just with one more mastering assignment for next week over the vessels. And that would also leave you with the quiz. Remember, there's always a quiz due the day before the exam, and that quiz will be over, thank you, that quiz will be over, again, the material to help you get ready for this test. Any questions logistically, what's going on? Remember too that next week's in your 11th lab, that pre-lab is extra credit, that your lab quiz is extra credit that night. If you missed a lab along the way and didn't make it up, then it will replace that low grade. If you've gone to all 10 labs and done well, then the points you receive in your 11th pre-lab and your 11th lab quiz will be total bonus points into your point total. And there will be a 12th lab quiz over the reproductive quizzes that are reproductive systems that will be given to you at your last lab. So there's actually 15 points of bonus points coming your way. That's 1.5%. And that's a nice little healthy boost for those of you who need that little kick in your grade. So, any other logistical things before we get started? Doing okay, Karen? Mm-hmm. Okay. What are we having?
What I have some extra Rice Krispies.
Some Rice Krispies. Well, okay.
Mixed it up this time, it's hard.
Okay. Okay. Let's look at 83, okay? 83. Sign, like a sinus, right? A cavity, a space, a recess. Sinistro, left. Do you remember what right was? Back in the D's, dextro. So dextro is right and left was sinister, is sinister. Cis, a state or condition. So in dialysis, you are going through a state or a condition where they are separating across, across the membrane. So people who are undergoing dialysis for kidney failure. You know that somato and soma both refer to body. Sphygmo, pulse. More specifically, when we get to blood pressure, you've all seen a blood pressure cuff, right? But the part of that instrument that measures the pulse is called the sphygmomanometer. Spiro and spire both refer to breathing. Respirometry, respire. We'll be doing a little bit of breathing exercises and talking about the respiratory system a lot more coming up soon. Squamous, remember we talked about flat, scaly cells, squamous cells. And steno is a narrowing. If you have a narrowing of your pyloric sphincter, you would have pyloric stenosis, or a narrowing of your trachea, tracheal stenosis. So any kind of narrowing, steno. Sterno, sternum, breast. Stomy, a surgical opening. So a tracheostomy would be the opening of the trachea. You know that skeletal and cardiac muscle are striated. They have striped, so stria referring to stripe. Stasis, standing still. Homeostasis, standing still. Hemostasis, when blood clotting, I'll use that word again today, but when you stop your blood from flowing, that's hemostasis, when you have a blood clot. And then under, subcutaneous, sublingual, we've seen that term a lot. You know that the brain has grooves called a sulcus. You've also seen a sulcus around the heart, the coronary sulcus. So any kind of groove or furrow, sulc. Super and supra both refer to above or upper, and sim and sin both together or with. Remember the pubic symphysis, where the pubic bones grow together, or to synthesize something. We've seen those terms many times. And I think I'll finish up with telo today. Systole, when the heart is contracting, that's considered the systolic pressure or the systole. We'll talk about that today. Tachyfast, someone with a fast heart rate is said to be tachycardic. Tarse, you know the tarsals are the bones of your foot. Now, that's a little bit of a misnomer. It's really the bones of your what? In a way, ankle in a way, right? But the foot bones. And then finally, telo. We saw telophase, last phase of mitosis, and we've also seen telodendria, the little tree-like branches at the end of the axon. So all we're going to do is go through 1, 2, 3, 4, 5 more. We'll do that on Tuesday through uni, and that vocabulary will be part of exam number 5. Now, is there anything that you can articulate of concern related to blood? And I'll just flash forward through this. We talked about the components of blood, how we can spin it down and we get the red cells and the buffy coat and the plasma layers. We talked about the 5 or so different functions of blood. And then we talked about the components of plasma and the different proteins that are part of your plasma, the albumins, globulins, fibrinogen, and the regulatory group. Talked about the things that are dissolved in your plasma, and then we started speaking more specifically about the different formed elements. We went through some features about erythrocytes, talked about hematocrit, talked about hemoglobin, and then after we finished up our conversation about red blood cells, we went into a little conversation about white blood cells, mentioned about antibodies and blood typing, introduced the different types of white blood cells, the granulocytes versus the agranulocytes, and that brought us to platelets. Now let me just back up, okay, just a little bit and ramp us into this, and go back to just this conversation about white blood cells, leukocytes. Anybody know why they're called leukocytes? White, right? White, leuko. Now, when you see them under the microscope, they've been stained. There has been stain added to the blood sample so that the cells appear kind of purplish or blue or red, right? That's not the normal color of a leukocyte. Why do you think they picked up the name leukocyte, white cell? We know red cell, right? Red blood cells have hemoglobin, they're red.
When you have infections and they use the pus.
There you go. That's exactly right. So when you have pus, right, that pus is really your body's ability or fighting off infection, and all that white stuff are the white blood cells that are coming to kill off the bacteria that are causing that infection. So pus is exactly where it gets it from. The white blood cells are the major component of your pus. Okay. So we've got 5 different kinds of white blood cells. Now each of these cells is specifically designed to do something very different, all related to your immune function. So remember that 3 of these white blood cells are called granulocytes. They are named so because they have granular appearance, and those granules are actually little vesicles filled with specific immune functioning proteins. For example, not to memorize right now, but neutrophils are specifically designed to attack bacteria. So if you have a bacterial infection, your neutrophil level will go up and you see pus, right? This pussy fluid. Eosinophils, they're specifically designed to fight off parasites, worms. So if you get a parasitic infection, and these cells would be elevated as they fight off your parasitic infection. Basophils, I told you last time, were the cells that release histamine. If you're having an allergic response, likely it would be the basophils that would come running to the rescue and would, as part of their function, release the histamines. And that would cause you to sneeze and have watery eyes and runny nose. So those are the granulocytes. On the bottom are the agranulocytes. These include the lymphocytes and the monocytes. They don't have the granular appearance to them. Now, all I told you to know were just a couple of key facts about these. You won't have to recognize them. I won't say what is that kind of white blood cell. We'll deal with that next semester. We'll learn a lot more about white blood cells and what's going on with those next semester. But for now, just know the following facts. Neutrophils are the most numerous, right? NN. Neutrophils are the most numerous of your white blood cells. Number 2, on the bottom, basophils, their numbers are in the basement. They are the least numerous of the cells. Thirdly, basophils are the ones that make histamines. I think that's just a nice little fact to remember. So we have the most numerous neutrophils, the least numerous, the basophils, and oh, by the way, these cells produce the histamines. That's it for now. Over on the white blood cell, on the lymphocyte side, I want you to remember, because we've been talking about blood typing, and in the blood typing conversation, we talked about antibodies. And where are these antibodies coming from? Your immune system is producing these cells, and these molecules, these antibodies. Remember, the antibodies are the second most abundant protein in your blood. They are part of that globulin group. And the cells making these antibodies are your B cells. They are a particular type of lymphocyte. You also have T cells. T cells have the ability of directly, some of them can directly attack something that's invading your body. And there are other types of these called natural killer cells. There's a whole bunch of them. And you'll learn some more of those facts at a different time. Then finally, there are the monocytes. These are monstrous in their size, monstrous, and they're the biggest in size. And these are the cells that will become your macrophages. Now, throughout your body, in different places, in different tissues, there are different types of macrophages. They have different names, but they all do the same thing. They chop up foreign things. They chop up dead and dying cells. They destroy debris that would come into your body. They might be going after bacteria. So they all do the same thing, but they may have different names throughout the body. We'll learn more of those different macrophage names, but they're all derived from these big white blood cells called monocytes. So as you put this together, just remember that all of these different white blood cells are involved in your immune response, whereas all the red blood cells were involved in carrying oxygen and CO2. Totally different functions, but part of your blood. Any questions so far on anything I discussed last time or that I reeled through quickly related to blood, its functions, plasma, about red cells or white cells?
With the percentages, how much are you going to be heavy on the percentages?
Thank you for asking. The question is, do we need to know about the percentages? I want you to know not the percentages of the white blood cells. I do want you to know that neutrophils are most numerous and basophils were the least. But as far as the actual percentages, no. Okay? So for now, neutrophils are the most numerous, basophils in the basement, the least numerous. When it comes to the other numbers, maybe that you're referring to, the lifespan of the different cells, I do want you to know that red blood cells hang around for 120 days. Platelets in a moment you'll see hang around for about 8 to 10 days. I want you to know that number.
Okay.
I want you to know the relative abundance of these cells. Every drop of blood, every microliter would have millions of red blood cells. Would only have a few thousand of white blood cells and would have hundreds of thousands of platelets. Not to memorize the numbers, but just have a relative idea of their abundance. Okay? So millions of red blood cells versus a few thousand white blood cells versus hundreds of thousands of platelets in every microliter of blood. Finally, the last number you need to know, a red blood cell is 7.5 microns in diameter. That's an important number to keep in mind, 7.5 microns. That white blood cells were bigger, 1.5 to 3 times bigger, and that platelets are smaller than 7.5. So I'll look at that number with you in just a second. Other question?
How long did you you said platelets 8 to 10 days?
8 to 10 days, and that's
What
coming
did
up
you
right
say
now.
before that?
Red blood cells, 120.
Okay, thank you.
And there's a table. And all these facts that I've just told you are all scattered in your PowerPoints. And there was a table that I gave you probably in the middle of the last presentation that gave you the size and lifespan of each of those different cells. Okay, I know they're kind of hard to see, but it's in there for you. Now, platelets, let's just turn our attention to these last little bit of our formed elements. Platelets are very small. They're about 1/4 the size of a red blood cell. So they're in the 1 to 2 micrometer range. And they're not cells at all. They are just little fragments, little blebs of a bigger cell that is found in the bone marrow called a megakaryocyte. Megakaryocyte. So tell me what this cell must look like. Megakaryocyte. Huge. Huge what? Karyo. Nucleus. Right? So these are cells with a very large nucleus. I'll show you a picture in a minute. But they're found in the bone marrow. Now, another name for the platelets are thrombocytes. Now, that's not a very good word for them because they're not cells. But you will hear them referred to as thrombocytes. Thrombo means clot. So cells that form the blood clots, thrombocytes, but they're not cells. Right? They're just little fragments of megakaryocytes. Now, their job is to help form a blood clot. And remember, I described to you fibrinogen. Fibrinogen was the protein floating in your blood. It was the third most abundant protein in your blood. And I told you that when a blood vessel becomes damaged, fibrinogen is cleaved to become a smaller protein called fibrin. But fibrin is sticky. And fibrin starts to form the actual clot. The platelets also are sticky. So the platelets and the fibrin and all the cells now form this blood clot, right? And that's what these cells are all about. They're all about forming blood clots. But there's a balance here. And I want you to think about this just for a second. You want your platelets to be sticky when they need to be sticky. But you wouldn't want your platelets to be sticky normally. And some people have sticky platelets. And what happens is that just a little bit of damage can start to cause blood clots. Just a little bit of damage to the inside of your blood vessels can cause this platelet progress, this platelet cascade to occur. So there's a really fine-tuned balance here that we'll deal with next semester. But it's really cool. We want these platelets to be there all the time so that at the very moment we need them, we have a blood clot being formed. But there's also a very tightly regulated balancing act here that we need to keep in mind. And we'll discuss that more. Remember that fibrinogen, the bigger name, is the protein in your blood that is soluble, not seen. At damage, the fibrinogen is cleaved to a smaller protein, fibrin. This protein is insoluble and helps with the halt of blood. Now, this term for halt of blood, I think I mentioned it last time, but let me say it again. To halt blood flow is hemostasis. Hemo, blood, stasis. Just so we have stasis in our vocabulary, standing still. So when your blood stands still, right, hemostasis, you have formed a blood clot. That is not to be confused with homeostasis, right? That's the term we've been talking about since day one. Homeostasis, keeping your body systems as closely in check as possible. Hemostasis, just referring to blood. Here's that number, about 120,000 up to around 300 or so thousand, not to memorize these numbers, but just an appreciation of their relevance to each other. And this would be the number of these little tiny fragments per microliter of blood. They're going to hang around for 8 to 10 days. So they're short-lived. If you had a reduced number in platelets, you would be perhaps diagnosed as having thrombocytopenia. Right? Thrombocytopenia, a deficiency in the number of thrombocytes in platelets. What would this individual's problems be? If you had thrombocytopenia, what would your symptoms be? Say it again.
You have like a bleeding disorder.
You would. It's not the same as hemophilia, right? It's not like you have a bleeding disorder where you don't have your clotting factors, which is what the Royals had in hemophilia. But you would bleed out. You would have more bleeding when you were cut, and you might bruise more easily. every day, as you go around your daily life, you bang your hand on the edge of the bedstand or, hit your steering wheel or whatever, and there is going to be micro fractures or little breaks in your capillaries, and your blood is going to clot very, very quickly, and you're not going to see any bruising typically unless you really banged yourself. But look at Uncle Joe's, Uncle Joe, right? Older guy or whoever, and their hands, they always seem to be kind of beat up and bruised up. Every time they hit just a little bit, those little vessels are bleeding a little bit, and their clotting isn't as good, and so they bleed out more. So you see this bruising, and you see this darkening blood that accumulates in their, where they've banged themselves up. Just in their everyday life, they'll have more bruising. They would probably have a reduced number of platelets. They'd have some sort of thrombocytopenia. And our numbers do drop with age. So at some point, we do bruise more easily. Now, this is just a cartoon representation on the right of a megakaryocyte. And what you see here is a cell with a pretty big nucleus, it appears, that is constantly blebbing off little pieces. Those little pieces are irregular, and those are the platelets. These cells, megakaryocytes, are found in the bone marrow. So it's just showing you that they're coming, in this case, from the pelvis, and these cells would be in the bone marrow. I wouldn't ask you to recognize this semester, but that is a megakaryocyte, and that's the cartoon representation of it, making the platelets. If you took a smear of your blood, okay, do just like you did in lab, take a little lancet, poke your finger. You could take that blood and put it onto a microscope slide. Now, what it doesn't say here is that in the steps here, they forgot to make a step, but they added some stain, right? You're going to add a little bit of stain. That's going to help color the cells differently so you can see them better under the microscope. You would then smear the cells out, just kind of smear the blood out along the microscope slide, and then stick it under the microscope. You would see something like this. The vast majority of what you're going to see are going to be red blood cells. Okay, all over the place, just red blood cells. And then, because of the stain, you would see darker, larger staining cells. These 4 cells are just some of the different types of white blood cells. As you see, they're in far less number. So they're relatively rare compared to the mass number of red blood cells. And notice that they're bigger. They have a bigger diameter than red blood cells. And then, boy, you almost have to have them point to it, but right there, they claim there's a little platelet. Just a little tiny bleb. And if you look around, you might see a couple other little blebs. Those would be platelets. So collectively, the red cells, the white cells, and the platelets are called the what? Formed elements, right? The formed elements of your blood. Back to hematocrit. If you take your blood and spin it, what's on the bottom? All red blood cells. What goes in the middle? Buffy, that's what goes there. What cells? What formed elements? The platelets and the white cells. And we call that layer where the white cells and the platelets go the Buffy coat. Right? So the Buffy coat is the name of that layer, strange word. That's the name of the layer, and in that layer is where the white cells and the platelets pellet out when you spin the blood. Just a quick table, again, if you like tables, fantastic. It's just a reminder that there are 5 different kinds of white blood cells: neutrophils, eosinophils, basophils. Those were all granulocytes, and then the lymphocytes and the monocytes are the agranulocytes. The numbers are over here. Nothing you need to memorize right now, okay? Again, nothing on these tables that I ever show you. If there's anything in the fine print, there's nothing you need to know from the fine print. The things you need to know are the things I've already included in the big print on the slides. Here's an electron micrograph of a blood clot. So what you see here are all these proteins are fibrin. The fibrin has gotten sticky. It's stuck to itself, and it's sticking also to the sticky platelets. You see all the little platelets aggregating. And in this clot, also you see red blood cells that are getting caught up, and surely there'll be white blood cells in there too that are getting caught up in this clot. All of your formed elements, remember, are made in your bone marrow. In fact, all of your formed elements are formed from a stem cell, a single type of stem cell, And that's shown in very small print up here as the hemocytoblast. Okay, so that's the one cell from which all of your blood cells are formed. It's found in your bone marrow, and under different hormonal and chemical signals, that hemocytoblast will go one of two directions. It will form what's called a lymphoid stem cell or a myeloid stem cell, and they're listed down here, myeloid or lymphoid. This is easy. It looks more complicated than it is, but for what I want you to understand is this. If the cell goes to the lymphoid line, over to the right on this image, only thing made from the lymphoid line are in fact the lymphocytes. So the lymphoid line makes the lymphocytes, period, that's it, the T cells and the B cells. If it goes to the other direction, if it goes to the myeloid side, it's going to make many others, myeloid many, multi. So the myeloid side is going to make everything else. And over on the myeloid side, you will see now, after many steps, there's a lot here, but erythrocytes are made from myeloid stem cells. Megakaryocytes, which then make platelets, are made here. And your other four leukocytes, the neutrophils, eosinophils, basophils, and monocytes. So multiple cells from the myeloid, only lymphoid line makes the lymphocytes. You may hear someone having myelogenous leukemia or lymphoma of some sort, different kinds of leukemia. Now, leukemia is a cancer of the blood. And just like any other kind of cancer, a cell has gone crazy and is growing uncontrollably. Now, it doesn't have to be the red cell or the eosinophil. It could be any cell in this very complex array of cell types. And based upon, you'll hear, like, again, a myelogenous leukemia, it would have to be a myeloid type, or a lymphoma or some sort of a lymphoid leukemia would be something involving the lymphocytes. And what happens is that these cells begin to grow uncontrollably and they get stuck, and that means that the mature cells are being crowded out, and sometimes the cells aren't even finishing their development. So you are lacking some important blood cell. Okay? All sorts of types of leukemia. Now, what sort of doctor would you see if you were having blood issues? Hematologist, right? So a hematologist would be a blood specialist who would deal with all sorts of things, from sickle cell anemia to leukemias to all sorts of other blood disorders. Last slide. Oh, let me go back. At the top of the slide, the word up here, hematopoiesis. Hematopoiesis. Hemo, blood, poiesis, formation of. So the process of hematopoiesis is the formation of all of your blood. You might also see it called hematopoiesis. They are the same thing. So you might see hemato or hemo. Don't get stressed out, they're the same thing. Just different folks will use different terms. Hemo or hemato. Formation of all of your blood. More specifically, I could talk about erythropoiesis. That would be just the formation of your red blood cells, right? Erythropoiesis. And we talked about this a little bit. Red blood cells are being made by the millions every second. And remember that red blood cells are only floating around for about 120 days, and that they're being broken down by your liver and by your spleen and recycled. You're making about 3 million more red blood cells every second. Just astronomical numbers. That means you're losing that many cells every second as well in your normal homeostasis. Again, red blood cells begin off as a myeloid stem cell, and it takes about 5 days for them to get formed. So if you were to fly out to Denver today, and you were feeling kind of cruddy because of the lower atmospheric pressure and you weren't getting the oxygenation that you wanted, you would start feeling better in about 5 days, because it's going to take that long for your body to up the number of red blood cells. Now, if instead we wanted to talk about just the formation of your platelets, we would use the term thrombopoiesis. And if we were dealing with just the formation of your white cells, we would instead use leukopoiesis. Okay? So hematopoiesis, all of it, erythro, leuko, or thrombopoiesis, referring to more specifically the formation of one of the formed elements. Questions on blood? Anything at all? Yes, ma'am.
This might be off the subject, but if a person, like, gets their spleen removed.
Yes. So if the spleen is removed, then they would no longer be filtering out their blood cells quite as well. The liver still does it, though, and can make up for most of it. The spleen is a place that helps to form and maintain your immune system. So that person might have a little bit of a reduced immune function and would have to be a little bit careful about exposing themselves to some things. But, and the spleen is, it's an important organ, but we can get around it. Okay, some organs we can't get around. You can't get around not having your kidneys, your liver, your pancreas.
If you're 88,
Say it again?
if you're 88 without a spleen, is there like, yeah, I know, my uncle, he had to get a spleen. When he was
Okay.
88.
Oh, 88. Oh, okay. I was hearing ADA. Yeah, 88. Yeah. I don't know if age is related to that or not, but the spleen is very, I'll say kind of, they call it friable because it's very fragile. If you start to take it out, it kind of just crumbles. It's a very oddly arranged organ, and but yeah, we can do okay without a spleen. Yeah.
So what happened?
Go ahead. Hand here and then question there. Go ahead.
Can you go out with only going with one kidney?
Absolutely. We'll talk about the kidneys next time, but people donate kidneys and do okay with one kidney all the time. And I'll say it now and I'll say it again next time. But you can live with about 20% kidney function.
Okay.
So a person can lose a kidney through donation or gain one kidney and have a 50%, half a kidney, if you will, 50% function and do just fine. It's not until you get down to less than 20% of kidney function that you would even be aware that you have a problem. Yeah, question?
I was just wondering, my uncle's spleen burst, so what is that doing to that?
Well, there's a couple of things that could have happened, but the problem is the spleen is such a vascular organ, right? Because it is, you're filtering your blood cells. So the spleen is getting a lot of blood flow. So to hemorrhage or to have the spleen get damaged can be a big bloody mess, for sure. A very big hemorrhage. Don't know specifically, but... Okay, any other questions about the content? Anything at all? Okay, let's jump over to the heart. I think you're going to feel really comfortable with this. You've already been quizzed on the flow of blood through the heart. You already know your chambers, your valves. You know the major vessels coming in and out of the heart. You are aware of some of the vessels carrying blood around the body. And what I'll be sharing with you will be a quick recap of what you know. Remember, this is chapter 18. I apologize, my numbers are still off. But much of what I'll be telling you will be a recap, and there may be a few things, well, there will be a few things along the way that we've not yet discussed in lab. So those are the things you really want to perk up and listen to. The rest of it, I hope that you'll sit back and say, okay, I got that. Confirmed, confirmed, confirmed. So let's take a look. Here's something we didn't talk about specifically. In an average-sized adult, the heart's going to beat, on average, 75 times per minute. Now, that is probably a higher average than the people in this room. That number is going to represent all individuals. And as we get older, our heart rate will go up a little bit. And so as a younger group, probably our average heart rate is a little bit lower than 75. But 75 times a minute is equivalent to 4,500 times an hour or 108,000 times a day. This is an amazing machine, right? The number of times our heart beats in a lifetime is just astronomical. It's really a cool thing to imagine. Now, each time the heart pumps, or each minute that the heart pumps, it is going to pump on average about 5¼ liters of blood. And you only have about 5¼ liters of blood in your body. So what this suggests is that with every minute, your entire blood volume has, on average, cycled around the entire body. So if you were a little red blood cell and you were grabbing onto a ride, sometimes you would go out the aorta and only go to the heart and back. And that would be a shorter, circuit. But other times you would jump the ride to the brain, and that'd take a little longer, or to the big toe, maybe a little bit longer. But on average, 1 minute, right, for your blood to circulate all around your body. We know that there are blood vessels that do all this work, and that those blood vessels are called arteries and veins. The arteries are always, by definition, carrying blood away from the heart. No exceptions. That's definition. And by veins, always carrying blood back toward the heart. Arteries usually carry red blood, but we know the exception. In fact, there are two exceptions. There's the pulmonary arteries that we know are blue, right? The pulmonary trunk and the pulmonary arteries. And during your development, your umbilical artery would have also been blue, right? The umbilical artery and the pulmonary arteries are the blue exceptions. Likewise, veins usually carry blue blood, blood lower in oxygen. Again, the exceptions would be your pulmonary veins, which are bright red, and the umbilical vein, which would have been bringing blood into the baby from the placenta through the umbilical structures. Now, there are arteries and veins referred to as the great vessels. You already know them. They are the great vessels because they are directly entering or leaving the heart. These are the vena cava, right, superior and inferior, the aorta, and the pulmonary trunk. When you do your heart dissections, I'm going to say next lab, but for some of you it's 2 labs away, but when we do our last lab and we do the heart dissection and the lung dissection, we're going to have full-size hog hearts, and they're big. And I think they're similar to human size. And I think you'll be impressed by the size of the aorta and the pulmonary trunk. I mean, it's about a 1-inch garden hose coming off. It's a good size vessel. Those are the great vessels. Now, the characteristics of the heart. You know that the blood, ideally, is moving in a unidirectional way. It's going through the chambers. There are valves along the way that are going to assure that blood does not go backwards. But occasionally, a valve will be a little bit leaky, and blood will go backwards. And clinically, what do we call those? A murmur, right? M-U-R, M-U-R. A murmur is a backflow of blood. A lot of people are born, they've got a slight murmur, and maybe they have that murmur all their life. And the doctor just says, you know what, you have a little murmur. We'll just watch it. And it's not usually a life-threatening condition, but if that backflow starts to get worse, then that means, right, if you've got too much blood going backwards, that means you don't have enough blood going forward. And that murmur can become a problem and can worsen and can necessitate surgery or other options if that becomes a bigger problem. Now, the heart is really like 2 pumps sitting side by side in your chest. They work together, but they also have an independence to each side. You know that the right side is not as muscular. The right side's job is only to pump the blood over to the lungs. So it's not as muscular. The right ventricle is not as impressive in its myocardium. But the left side is much more muscular, much more impressive, and it has to squeeze blood with higher pressure all the way to your brain and to your big toe. So it is a bigger workhorse of your heart. There has to be pressure, right? This idea of blood pressure. We'll talk about blood pressure next time when I go through the artery and vein review with you, and we'll spend some time discussing blood pressure. But there has to be enough pressure to get the blood up to the brain and enough pressure to get the blood down to the toes, right? And maintain that pressure along the way. And we'll talk about how the muscular arteries are going to maintain that pressure next time. And we'll also talk about the blood pressure as it goes through the heart a little bit today. If there isn't enough blood pressure, then the blood can't sufficiently get to the organs. So if your blood pressure is low, really low, you're going to faint, right? You're not getting enough blood to your brain, or you're not getting enough blood to your other organs. And one organ that I'll tell you right now, besides your brain, that gets really pissed off when it doesn't get enough oxygen are the kidneys. And we'll mention that again next week. But the kidneys are really, really finicky and don't like having less oxygen. So a person who's having heart failure, who has very low blood pressure and the heart's just not keeping up, not only will they feel lousy and not have any energy, But their kidneys may start shutting down because the kidneys just simply require constant blood. Because what are the kidneys doing? They're filtering the blood, right? So if you're not pushing the blood through the kidneys at enough pressure, they're going to get really upset and kind of just say, forget this process and shut down. We'll talk about that next time. Now, there are two circuits. These arteries and veins in your heart are working to create two circuits. You know this. The pulmonary circuit is dealing with just the valve, or just the vessels, bringing blood to and from the heart and the lungs, right? That's the pulmonary circuit. Remember, that one's all screwy as far as color. The blue and red's all reversed. Then there is the systemic circuit, and that's what the left side of the heart's doing, and that's the one sending blood to all of your, quote, systems. You know this story as well. You know that oxygenated blood, red blood, is going to leave the left ventricle. It's going to go up through the aortic semilunar valve and enter the aorta. That aorta is going to go throughout the body, and as it branches out, it's going to get smaller and smaller in these different arteries that we're going to name. And eventually, those arteries will get really small to what we call arterioles. The arterioles will eventually drain into capillaries. The capillaries are very thin, delicate vessels. I don't want you to think of capillaries as just small arteries. And I think I know at one point either I was taught that or I just conceived that capillaries were just miniature arteries. I don't want you thinking that because arteries have 3 layers that we'll talk about again next week. Capillaries only have 1 layer. So capillaries are not just minuscule mini arteries. They are a different kind of thing. So the arteries eventually go to the capillaries. The capillaries are where all the gas exchange happens, but not only gas exchange, but also nutrients are dropped off. And this is where hormones will enter into the tissues. And this is where waste products will be picked up. So, and this is where the lymphatic fluid exchanges with the cardiovascular plasma. So all of the exchange between your blood and the tissues is going to occur at these very thin vessels called the capillaries. After that exchange happens, the blood will then continue its journey out of the capillaries and into veins. And those veins will then eventually merge back to our heart, getting larger and larger along the way, and eventually draining back into the inferior vena cava, if you're coming from the legs and the trunk, into the superior vena cava, if you're coming back from the head, the neck, and the arms, or back from the coronary sinus, if the blood has only gone to the heart muscle. And then around and around and around we go, right? Over and over and over and over again. Now, did Mr. Mueller describe to you a square heart? I don't think he does a square heart thing, right? So that's okay. When I'm describing the heart, I kind of draw out a square heart. It's because my artistic skills, as you well know, are horrible. I struggle with stick figures. For me to draw an anatomically correct heart, like this route that's up on the screen, really is very difficult for me to do. So I always start off in lab by showing people a square heart. And all it is 4 chambers. And I put the 4 chambers and the 4 valves. And it's just a different way of presenting the heart in a easier to draw version. If you had seen the square heart presentation, I'm now going to beg you to move away from it and start thinking more anatomically correct. You should now be able to, after having been through lab and thinking about the blood flow through the heart, you should be able to take your finger and basically trace through an image like this. And as you're going through this image, notice what chamber are you in, what valve are you passing through, what color is the blood. Everything on the right side is blue, everything on the left side is red. You should be able to kind of follow through this, know where the aorta, the pulmonary trunk, the inferior and the superior vena cava are. So you need to be able to go there. And I'll help you with that today as we continue through this conversation of the heart. So anatomy of the heart. Okay, it's a relatively small organ for its importance. It has a conical end that we call the apex, the little cone end. It's not a bad estimate to say it's about the size of your fist. It is going to be located a little off-center in your body. It's in that area called the mediastinum, between your lungs. protected well by your sternum and your ribs and your vertebra from behind. It's going to be a little bit off-center, though, such that, and I'll show you a picture in a moment, that the heart is twisted just a little bit. And let me get to the next slide, and I think it's easier for me to describe it. At the bottom, oops, the heart has that little pointy area, the apex, right? And the apex will actually be slightly antero-inferior. What does that mean? It'll project a little bit toward the front and a little bit downward and off to the left side. So the heart sits in your chest a little bit off to the left with the conical section, the point going down to the left, but then kind of coming out a little bit outward. Okay. And that means at the top of the heart, what we call the base of the heart, is more posterior superior. So is kind of twisted, a little bit twisted toward the back, and the top part of the heart is called the base. Now, what is it that comes in and out of the base? Yeah, the aorta, the pulmonary trunk, and the superior vena cava all come down or enter into or leave the heart at the top, what we call the base of the heart. Okay. So let's just take a look at the heart in its anatomic position. Notice, right, it's a little bit off-center. It's a little bit shifted to the left. There's the apex. And if you could appreciate the twisting of this, that apex will be pointing a little bit toward the front. The base, the top of the heart, right, is where the pulmonary trunk and the aorta and the superior vena cava are coming in and out of the heart. This is the diaphragm. And notice how beautifully protected, at least ideally in this cartoon, the heart is protected by the sternum quite nicely. And even the major vessel, the aorta, comes up, and those three branches off the aortic arch are nicely, ideally protected right behind the manubrium. And the aorta, as it comes up and over and comes back down the posterior side of the heart, extends down a little bit and is even protected more by the xiphoid process. Okay? Now, that xiphoid process can be a problem for ball players or other people with some trauma, but you'll hear about every year, unfortunately, some infielder, baseball or softball infielder, usually a pitcher, who the ball comes right back at them, boom, and hits them right in the chest, right at the wrong angle, and snaps off that xiphoid. Now, for many people, the xiphoid's kind of round, but for some people, it's a little bit longer and a little bit more prominent, a little more pointed. If you look around upstairs in the lab, the two real skulls or skeletons that we have, you'll see that the xiphoid has quite a bit of variation in its shape. But if you were to snap that little xiphoid off and it was already kind of pointy, you could imagine severing the aorta. Now, the aorta is a very muscular structure. I mean, it's a nice, thick garden hose, but you could, with the right pressure and all the wrong situation, sever the aorta, and that person's going to drop. There's nothing we can do for that major hemorrhage. So now, what do little kids wear? Right? Little pitchers will wear little chest protectors, so these little strap-on thing or a built-in chest protector into the shirt, and that's just going to dissipate the energy. You don't see major pitchers wearing those things. You don't see high schoolers even wearing those very often. They're usually in smaller kid sizes. And this is why, Little League keeps changing the darn bats on us, because they realized that the ball was snapping off those bats was too much power. And the little kids weren't able to react, and too many kids were getting hurt by these balls coming flying at them at high trajectory and velocity. So they've been changing up the bats a little bit, trying to slow them down a little bit, and encouraging some infielders to wear these pads or these little guys. You've heard about it, probably. It's unfortunate. Okay. This is the actual heart. Now, I wouldn't ask you to label these cadaver images. One thing you might note is this was a smoker, right? Nice black lungs. The other thing you'll notice on this is, here's the heart, right here. Here's the diaphragm. Two major vessels coming out, 1 and 2. Now, you really can't see where it comes from. The one right here, that's the pulmonary trunk. It's the more anterior of all the vessels. And then if you weren't sure, here's the aorta, and watch how it goes up, and there's the first branch. What's that first branch of the aorta called? Off the aortic arch? Brachiocephalic trunk, right? And what is that brachiocephalic trunk going to do? It's going to go up, brachiocephalic. It's going to go to the right arm through via the subclavian, and it's also going to continue up the right neck, the right common carotid. The second branch, there it is, coming off the aortic arch, goes right up the left side of the neck. That is the left common carotid. The third branch you can barely see on this would go off to the left arm, left subclavian.
What was that silver?
Oh, a probe.
Oh, okay.
Something you carry around in the lab upstairs, right? Little pointer. Yeah, that's not normal. Yep. Maybe that's what killed them, right? Someone left their surgical probe in there. No, that's just a big old pointer, okay? Okay, good observation. So let's take a look at this picture cartoon of the heart and lung. So again, you're going to see this when you open up your fetal pig again next week. Look at it again, and you're going to see the diaphragm. Now, for most of you, you ruptured or tore away the diaphragm when you cut into the abdominal pelvic cavity. But you'll still see it kind of hanging over to the side. Imagine that it's still intact. And you'll notice that the heart is sitting right there on top of that diaphragm. So here's the diaphragm, and the heart is attached directly to it. We'll see that again in a moment. Remember the lungs? We've got two lungs. Here's the left lung over here, and notice that the left lung is a little bit smaller. It has to make room for the heart. So the left lung only has 2 lobes in it. You'll see that tonight and next week in lab. The right lung has 3 lobes in it. There's nothing interfering with the heart. So the right lung is larger and has 3 lobes to it. Even further proof that we're not symmetric. Right? I mean, when I think about our body, I think when you think about our body, you think of this very symmetric thing. 2 arms, 2 legs, right? 2 eyes. But when you start looking inside, I think you should become more and more amazed at how asymmetric we are. I mean, we've got 2 lungs that aren't the same. We've got a weird heart. We've got 2 brains that we know aren't the same, right? 2 hemispheres. We've got 1 spleen, 1 liver, 1 stomach, 1 gallbladder. So there's a lot more of us inside that's asymmetric than we have mirror images of each other. So we see the apex of the heart. Remember, too, that around the lungs you have the serous membrane. So what was that serous membrane around the lung? The pleura. Remember, there are 2 layers to every serous membrane. The visceral layer was shrink wrapped to the lung itself, and the layer lining the thoracic cavity would be the parietal pleura. Remember this? And the heart, same thing. We said that there was a visceral layer, shrink wrapped to the heart, and a parietal layer. And we'll get to that more in a second. Now, imagine that we're going to slice this whole thing right across this plane, and then lay this down and look at this from the top. Okay, so we're looking at a cross-section of that moment before, and posterior and anterior are marked for us. This bone is the sternum. So right behind the sternum, there's the heart. In fact, the heart is attached to your sternum, and it's attached to your diaphragm. So through the pericardium. So the heart is really well anchored. Boom. It's right there. It's attached to your sternum. It's attached to your diaphragm. It's not going to wiggle out of place. You see the vertebra on here? What kind of vertebra would this be? It better be a thoracic vertebra since we're in the thorax. If that's a lumbar vertebra, our heart fell a long way, right? And my heart certainly isn't up in my neck, so it can't be a cervical. It's not a trick question, right? Just put it all together. That's going to be a thoracic vertebra. What would this tube right here be that's not labeled? Little flat guy right here. It's way posterior. It's right anterior of the vertebra. What tube would be passing down in that area? I'm hearing esophagus. That could be right. You might see that again. Go confirm it. I'm not saying right or wrong. I'm just saying go confirm it. Okay, you might see that. What is that thing again on a test? What else do we see here? 2 lungs. I see the aorta, right? It's kind of, we cut it open the aorta there. So that kind of gives you an idea. And you see some ribs coming around the outside as well. Okay. Now, the pericardium. Pericardium, when you think of the pericardium, what comes to mind? 2 layers, right? Visceral and parietal. We've been through this. Around the heart, good. The visceral layer shrink wrapped to the heart. The parietal layer lining what? What's parietal mean?
Wall.
The wall. No one ever asks me this in lab 1, but what wall is the parietal pericardium lining? The pleural, it makes sense, right? The parietal pleura is lining the is lining the thoracic cavity. The parietal peritoneum is lining the abdominal pelvic wall, right? What wall is the parietal pericardium lining? And it turns out the pericardium is a more complex structure than just a double serous membrane. There's actually a fibrous, leather-like sac around your heart. Okay, and that's called the fibrous pericardium. Never mentioned it before, but your heart is held in place by a fibrous and a serous membrane. Now I'll show you a picture of this in a moment. This pericardium, right, this leather sac, is going to restrict the movement of the heart, and as I've said, it is directly attached to your sternum and to your diaphragm. What I never told you before, because we didn't need to know it yet, is that the true leather-like sac is called the fibrous pericardium. It's the more outer layer of this thing around the heart. It's made up of tough, dense connective tissue. And the inner side, the inner pericardium, is the 2 layers that we're accustomed to, the serous membrane. So when I say parietal pericardium, the parietal pericardium is lining the inside of this leather sac. You picturing with me? Okay, so the parietal pericardium is lining the inside of this fibrous pericardium. Let me show you a picture of this and also remind you that the visceral pericardium is also called the epicardium. We saw that in lab, and I'll mention it again in a few minutes. But the outer layer of the heart, the epicardium, is the same as the visceral pericardium. If I took a little chunk out of your heart, there'd be 3 distinct layers, right? The epicardium on the outside, the myocardium, the muscle layer on the inside where the cardiac muscle is, and on the inside, the endocardium, and that's going to line the inside of your chambers and basically coat your valves. As we age, there's going to be a layer of fat deposited underneath the epicardium, and I'll show you that in a moment, this fat layer that accumulates. Let's try to get our mind around this pericardium, because I think I struggle a little bit with this sometimes, trying to think through this the first couple times. So I'm showing you a heart, and all this white stuff would be the fibrous, right, the connective tissue. Notice that this is the diaphragm, so the heart is directly attached to that through this fibrous pericardium. I'm going to zoom in. So they've already done this for us. So I'm going to zoom into this little area up here in this box and go into this enlarged area. So 3 layers around the heart. The outer layer, right, the tough white layer, that is that fibrous pericardium. That's the sac. Then, deep to that, lining that, in fact, this white layer, that is the what? Parietal layer. It's lining the wall of the sac. And then, right on the heart itself, this layer, that would be the visceral layer. And in between, just like we've always had in between these two membranes, there's always going to be a little fluid-filled chamber. So there's your serous fluid in between the two membranes that we've talked about in the past. While I'm here, does that make more sense? This idea of a fibrous pericardium, a leather-like sac around the heart, and that the parietal layer is lining the inside of the sac? Right? Because in the past, I'd always say the wall, and no one ever says, what wall are you talking about? Where is this parietal layer? What wall is it lining? Now we've got that mystery solved. Question?
So they're connected? The parietal,
It's 3 layers. So when you think of the pericardium now, think not of 2 layers, but of 3. Visceral, on the heart. Parietal, lining the inside of the sac. And then the sac itself. All connected. All connected. Question? The mediastinum is simply the anatomical name for the area between your lungs. Okay, media, middle, stinum, sternum, the area back behind your sternum in between your lungs. So we would say that the heart is within the mediastinum. Now, well, let me go back while we're here, and let me just quiz you on a couple of things. So, what chamber is this right here that I'm putting a big dot on? Boom, I like it. Left atrium. And what made you know that it was left atrium? There's a few ways. Give me one way you know it's the left atrium.
the friction of the apex.
Ah, you know that the apex is the left side. Good.
The wall is really thick.
The wall is very thick. That's definitely your left ventricle. One more thing for sure that tells me that's the left atrium. Coming in, I see red pulmonary veins, right, draining blood into that left atrium. So any of those ways would have gotten you your orientation and helped you appreciate that that was the left atrium. Now, what is this valve? Mitral valve, bicuspid, or the left atrioventricular valve. What are these muscles? Papillary muscles. And the strings are the chordae tendineae. This valve? The aortic valve, or the aortic semilunar valve, is going to go up out of the left ventricle and go a little bit behind and then come up as part of the aorta that we can't see here. Very good. That feel comfortable? So again, I think you know this, and I've just said this, but I got a little more information on here for you. So if I took a little chunk of your heart and I was looking at the wall of the heart, there would be 3 layers. Epicardium, outer layer. We know these prefixes help us. The epicardium, a.k.a. visceral pericardium. Very thin layer. It's just a simple squamous epithelium. Nothing you could really appreciate. It's just a very thin layer. The middle layer, the myocardium, muscle layer, cardiac muscle. If one has a myocardial infarction, they've had a heart attack. They've had a lack of oxygen to this muscle of the heart. The inside layer, endocardium, where the blood is touching the inside of the chambers and lining the valves. A person can have an endocarditis, an inflammation of the endocardium. That can be a really serious infection, in part because it can damage the valves. And to damage the heart valves is to really put a big, hurt on the heart. So endocarditis, a bacterial or viral infection that damages the heart inside, can be very, very serious. Here's a chunk of that heart wall. And just the outside layer, simple squamous, that is your epicardium. Here's that fat layer, the yellow that I told you accumulates with age underneath the epicardium. The thick middle part is the myocardium, that's your cardiac muscle. And then lining the inside would be the endocardium. That's just another connective tissue layer. Now, quick reminder, you've seen cardiac muscle under the microscope. You know some of its characteristics. Just review that with me. The muscle fibers of cardiac muscle would be, compared to skeletal muscle, shorter. Remember, skeletal muscle can be really, really long cells. So these are going to be shorter cells, shorter fibers, and they are going to have branches to them. Skeletal muscle was long, skinny cells. Cardiac muscle have shorter cells that branch. A lot of mitochondria, a lot of energy need in a cardiac muscle cell. We still have the fibers, the cells arranged in bundles like fascicles. But when the heart squeezes, when the ventricles squeeze, not only do they contract, but they also twist. And this will become important in a moment. But as the ventricles contract, they're going to twist. And that's going to push blood up and out of the ventricles, just like ringing them up. It's going to ring everything up and out through the aorta or the pulmonary trunk. Now remember that cardiac muscle is striated, just like skeletal muscle, has the striping patterns. And do we remember what those striping patterns are caused by? Striations are caused by repeating light and dark bands caused by myosin and actin, right? That whole sliding filament that we talked about a while back. We want the heart to act as a single unit. Unlike our biceps brachii muscle that can work little or more, we want the heart to be a very concerted, coordinated contraction. We want the two atria to contract simultaneously, and then we want the two ventricles to contract simultaneously. That lub-dub idea. You don't want all four chambers of your heart contracting at the same time, nor do you want the cells all doing their own thing on their own. That would be a mess. When the cells kind of just, everybody's doing their own business, we call that fibrillation. Right? When the heart just kind of quivering and all the cells are out of synchrony, we call that fibrillation. And we run down the hall and we grab a defib, right? Boom, shock them and try to get the heart back into a synchronous rhythm. We'll talk more about that next semester. Because we want the heart to contract as a single unit, we need a way for the electrical signal to travel very quickly from one cell to the next within the heart. And you've seen that special modification. That's the intercalated disc. So the intercalated disc is the electrical synapse. It is the place where the electrical signal can travel very, very quickly from cell to cell and assure that the chambers cooperate and work together. Now, just a quick reminder again, when you're looking at a cartoon of cardiac muscle, I think you're struck by two things when you look at this. One, you still see the striping, right? You still see striations. I see a branching here. So there's definitely some branching going on of the cells. Big old, there's some nuclei in here, and there's also some big old mitochondria that are quite numerous in here. Now the purpose of this slide is to tell us a little bit more about intercalated discs. Remember intercalated discs, when you look at cardiac muscle, I always say it looked like bamboo, and you had the striations, but you had these thick bands that kind of showed up along the way. And an intercalated disc is kind of a combination of two things that you've already heard about. It's part desmosome and part gap junction. Now we talked about both of these a long time ago in the skin chapter. And we said that epithelial cells are tightly held together by a few different ways. There were tight junctions and gap junctions and all that kind of stuff. The desmosomes were on that list. And remember the desmosomes were, I always called them little spot welds, little rivets. It's, you would find desmosomes on cells under a lot of stress. And they hold things together nicely. Well, I would argue that the cardiac muscle is a pretty stressful tissue. It's constantly having to pump, and the cells are constantly being stretched. And so there are desmosomes in the cardiac muscle, keeping the cells tightly adhering one to another. But there's also a gap junction. The gap junction idea was there are pores that allow for molecules, and in this case electrical signals, to travel directly from one cell to the next. So an intercalated disc, this really cool thing in cardiac muscle, is really a combination, a hybridization of a desmosome and a gap junction, holding stress tissue together, while allowing electrical signals to pass quickly from one cell to the next. You are not going to go back and review the Z, H, M, I, and Z stuff. No, right? Don't go back and worry about that. That was the story about the striations and the overlapping myosins and actins. Remember, though, that that is present in cardiac muscle. Cardiac muscle is striated. It does work the same way. There are sarcomeres, actins, and myosins that slide over, and the heart contracts. Remember that when you see an intercalated disc, right, it kind of looks like this little wiggle through here. These are the intercalated discs in this image. Now, part of this story I'm going to go through very, very quickly because you already know about the chambers of the heart and the valves of the heart and the great vessels. There are a few slides, though, here in the next 15 minutes that I will stop and focus on more carefully. And so let's just kind of start thinking about the heart. So we've got this four-chambered organ. There are two smaller atria, two larger ventricles. The atria are the more superior chambers. The ventricles are the more inferior chambers. The atria are very thin. They're rather wimpy compared to the muscular ventricles. And external to the atria, one would find these wrinkled, flappy structures called auricles. So each of the atria have external to them these two auricles. The atria, remember, are receiving the blood. The right atrium receiving blood from the from the systemic circuit, right? Blood's coming in, it's blue, it's already been used up, it comes in, and then the left atrium is receiving blood from your pulmonary circuit, blood that has come in freshly oxygenated from the lung. Atrium means entryway. Your home, the entryway, the church, entryway, is an atrium. Blood is going to come into an atrium and continue down to the ventricle on the same side, unless you're a fetus. Let's stop for just a second and think about what happens in the fetal heart. Blood comes into the right atrium, right? Where did that blood come from? As a fetus. Before you were born, you got your oxygen from mama. And the blood coming into the heart was already oxygenated. So it came into the right atrium and then jumped over to the left atrium through what hole? What's a hole? In anatomy, a hole is a hole, opening, foramen. And what shape is that opening? Oval. So the foramen ovale, and that's the opening between the two atria. Right? So what happens? You got oxygenated blood coming in. Fetus doesn't use its lungs. Right? Lungs aren't developed, lungs aren't being used for oxygenation. So the red blood jumps from the right side over to the left side. And then is pumped through the baby's body, through the baby's left ventricle. And then that blood leaves the baby, right, back to mama, and the placenta and the regeneration of that blood. So the vessel carrying the blood away from the baby's heart is an artery, but it's carrying blue blood, isn't it? The vessel that was carrying blood to the baby, things coming to the heart are veins. It was carrying oxygenated blood. So again, that's that red and blue exchange that happens in the fetus and in the pulmonary circuit. Okay, little bit of a break there because I wanted to make sure this statement wasn't oversimplified. Blood that comes into an atria continues down to the ventricle on the same side. That's in you and me. But in the fetus, right, we got that little jump over to the other side. There are the great vessels, two major arteries leaving the heart. On the right side, the pulmonary trunk, sending lots of blood over to the lungs. From the left side, the aorta, sending oxygenated blood throughout your entire body. So I'm going to just point to structures. I'm hoping that this labeling activity would be reasonably review and straightforward for you. So I point and you scream, okay? I put dots on it. How's that? If there was, let's say it's a green sticker. Ooh, let's do green. Okay, because we've had green stickers, right? So let's pretend, okay, green sticker right there. Right? The atrium would be the hole inside, right? The space inside. The thing, the flappy on the outside is the auricle. So that'd be the right auricle. Can you see that green? On that flappy, that's the left auricle. This portion of the heart is called the pointy is the apex. This vessel, aorta, it would be even more specifically the ascending aorta because it's the part coming up and out versus the top, which was the aortic arch. Okay, we got it. Let's talk about those three branches coming out the aortic arch. The first one, a little bit bigger. Brachiocephalic trunk. It is an artery. What happens right about here and here? It branches and goes to the subclavian and the right common carotid and the right subclavian. The second branch, left common carotid artery. Third and final branch, left subclavian artery. I'm going to get you, forcing you to think all the way through because on the practical, see people will just put subclavian. And I'm like, left or right, artery or vein. So just get in the habit of telling yourself the whole thing.
Do you have to put common in the title? You have to know the difference between when they have common and when they don't?
Yes, and the reason is, the question is you have to know common versus when it isn't common. Up the neck, you have the common carotid. But what happens up next to the side of the head? The common carotid splits into the internal and external carotid. So yes, I want you to use common carotid through the neck and internal and external carotid when you see the split at the head. Likewise, common iliac. As the aorta comes down, it splits into the common iliac arteries. And then the common iliac arteries split into the internal and external iliac arteries. So the same idea. The common split into internal and externals. Okay? And the other thing that's been a lot of trouble is this guy right here. Pulmonary trunk, which then splits into the right and left pulmonary arteries. So what comes, the pulmonary semilunar valve, blood then goes into the pulmonary trunk and then into the arteries, right? The trunk, tree trunk, big thing before it branches. What else do we have here? Down here, inferior vena cava. Up here, can you see it? Superior vena cava. And these guys coming over, one of these would be a pulmonary vein. All these arteries on the outside of the heart, coronary arteries, all the blue things are cardiac veins. If I had a sticker, I'll change colors for a second. If I had a sticker, what chamber, if I were to stick something into the heart there, what chamber am I going to stick into?
The right ventricle.
The right ventricle, whereas over here, the left ventricle. Stick it in here. Right atrium deep down and good, left atrium. Okay, so we're doing well in the heart. Now, you see the groove over here? There's a groove. That groove goes around the heart at a weird angle, all the way around, circumferential, all the way around. And that groove is called the coronary sulcus. Now, your book and other books will also call that the atrioventricular sulcus because it's the groove that separates externally the atria from the ventricles, right? So everything, over here, right, is atria, if you will, right? And everything below it is ventricles, if you can make any sense of what I just did. Now, let's, on the actual heart, I think you can appreciate this would be the pulmonary trunk. Pulmonary trunk here, and then the aorta. You see it kind of coming up and around, and there's that brachiocephalic trunk and the left common carotid coming off. You can also appreciate how deep that sulcus is and how big these coronary arteries are on the surface of the heart. I just said this. Now you've got it in words if you didn't catch it all. So the atria out externally are separated from the ventricles by the coronary sulcus, which is also called the atrioventricular sulcus. On the front of the heart, between the right and left ventricle, there is a sulcus with some coronary arteries traveling in it. That is the anterior interventricular sulcus. On the back side, there's a posterior interventricular sulcus. Underneath, pull the heart up and basically look at it on the lab. I think the post goes right through right about here. On the back side, what do we see here? This big blue vein. All the blood from the heart's coming back to it. The coronary sinus, absolutely. So all the blood from the heart's coming back through a bunch of cardiac veins, and they're all draining into that coronary sinus. That coronary sinus then drains back into the right atrium. What is this chamber? I see red things coming in. That's the left atrium. On your models, that's the back door that flips down. Right? The back door that flips down that exposes the left atrium. It kind of looks just like that. Okay, again, these are very significant. When you see your heart next week, I think you're going to be impressed by, again, how thick and muscular the aorta is. And you're going to notice how flimsy and amazingly wimpy the vena cava are. Now, the next few slides, honestly, are things that you are very familiar with. So what I'm going to do, actually, that's not a true statement. I want to introduce the fibrous skeleton, then I'm going to finish up and, come on. Let me just talk about the fibrous skeleton, and then we'll be done for the day. So there is in your heart, and you shouldn't be surprised by this, a skeleton. It's not a bony skeleton like you and I have for our bones, but it does give the heart its structure. You've got 4 chambers and 4 valves that somehow have to maintain their place within this hollow organ. So it's maintained by this fibrous skeleton. It is a connective tissue. It is going to physically separate the 2 atria from the 2 ventricles. It's also going to secure the valves in their proper anatomic place, and it's going to act as an insulation so that the chambers of the heart all don't contract at the same time. You don't want all 4 chambers contracting at the same time. You want lub-dub. You want atria, then the ventricles. So this fibrous skeleton also acts as an insulator in helping this. Now, this is a very idealized cartoon. I have never seen a heart cut that would ever look anything quite like this, but it brings the idea to you. If you were to cut the heart at this plane, basically I'm cutting it along the coronary sulcus, along the dividing line between the atria and the ventricles. I would, ideally, find a single plane where all 4 valves would be lined up. So here I can see all 4 of these valves. And the white stuff around it, that's the fibrous skeleton. So the connective tissue that's binding all this together and would be separating the top from the bottom chambers. Now, which of these must be the bicuspid? Don't look at the labels, just look at the pictures. Which of these valves must be the bicuspid? 2 flaps, right? 2 points. Cusp means point. Does that not look like one of your bicuspid teeth, right? I think this was a dental artist, right? It looks just like a tooth. That's a bicuspid. This, I understand now why they call it the tricuspid. And your valves do have 2 and 3 parts to them. I do not, nor have I ever understood why this would be a semilunar. That look like half a moon to you? Not me either, right? But the only valve I'd ever ask you to label here is the bicuspid, because you can see why it's called the bicuspid. It has 2 points, yes? It stands out as being different. Fair enough? Now, I'm not going to go through the internal anatomy with you right now, but I want to go back to the picture of the heart, and I want to point out something to you, and then we'll be done. And it has to do with the valves. Oh, I may not come to it right now. Speaking of tri and bicuspid, okay, I just want you to see this picture, and I'll come back to it. I'm going down, there it is. I'm going down 2 or 3 pictures from where you are in your notes. But just look at this image. Look at this valve. What is the name of this valve right here? Its nickname? Bicuspid. 2 parts. Look how many papillary muscles are attached to chordae tendineae. 2. Over on the right side, tricuspid. 1, 2, or actually no, 1, 2, 3. There's 3 papillary muscles that are helping to adhere those or pull down on those 3 sets of chordae tendineae, right? So you just kind of see that little bit of a difference between the right and left chambers and the chordae tendineae and the papillary muscles. Please review the flow of blood through the heart. And I won't review that with you unless there's any specific questions. On Tuesday, I will go through a little bit on what's left toward the end of this about the coronary cycle and about the angina and what a heart attack is. I don't have a whole lot more on the heart, to be honest. I did get a little bit further this morning, but I'll finish up this little bit with you on Tuesday, and then our conversation about arteries and veins is really not too bad on Tuesday. So I hope you get home safely, have a good weekend, get some good studying done, and I will see you all next week.
Thursday's the test?
Yes, the test will be next Thursday.